Good morning, everyone. My name is Geneviève. I'm an assistant professor at the University of Montreal in the Faculty of Pharmacy and at the Institute for Research in Immunology and Cancer. It is my pleasure today to host today's seminar that will feature two presentations from Dr. Annie Ciernia's research group at the University of British Columbia that will tell us about epigenetic mechanisms of transcriptional regulation involved in brain plasticity and neurodevelopmental disorders. I want to remind you that if you have any questions for the presenters during their talks or after their talks, please type them in the chat box. I will be able to relay the questions to the presenters after their talk. So our first speaker today is Jennifer Kim. Jennifer is a neuroscience PhD student at the University of British Columbia under the supervision of Dr. Annie Ciernia, where she studies how the immune and nervous system interact to impact brain function and behavior. Prior to joining the Ciernia's lab, Jennifer completed a master's degree in biology at San Francisco State University and an undergrad degree in biopsychology at the University of California in Santa Barbara. She then joined the Ciernia's lab for her thesis work, where she focuses specifically on how microglia, the resident immune cells of the brain, are epigenetically regulated in innate immune memory in the brain. So she will be talking today she will be presenting a talk today that's entitled Innate Immune Memory, an Orchestrated Microglial Response in the Brain. So Jennifer, you can go ahead and start.
Thank you for the lovely introduction, and thank you all for joining today and for the opportunity to present on some of the cool work that we're doing in Microglia in the lab. So to start, I'll give a quick primer on the specific brain cell type we focus on. So microglia are the brain's resident immune cells, and in their immune function, they constantly survey their microenvironment to respond to injury and inflammation and phagocytose cell debris. And in addition to their traditional immune roles, they also play a host of different functions in the brain, working with other cell types to regulate neuronal cell number, shape brain circuitry, and fine-tune neuronal connections throughout life. And on top of this, they make up 10% of all of the cells in the brain. So how do microglia tune their responses to the local environment? So much like a musician tunes their instrument to the perfect pitch to synchronize with the rest of the orchestra, microglia also fine-tune their responses to adapt to their microenvironment and take on specific phenotypes accordingly. So microglia have a very unique ability to adapt to a changing environment in a very signal-dependent way. And as the brain's innate immune cells, they're activated in response to inflammation within their vicinity. And an example of this fine-tuning microglial response is innate immune memory. So innate immune memory allows immune cells to adapt to an actively changing environment by either enhancing their responses to a secondary infection, also known as immune priming, or repressing their responses to a secondary infection, also known as immune tolerance. And disruption of this innate immune memory in the brain has been implicated in the pathology of a wide range of brain disorders, including Alzheimer's, MS, autism, depression, and schizophrenia. And while the mechanisms that underlie innate immune memory in the brain aren't as well understood, they do appear to rely on gene regulation within microglia. So LPS is a commonly used model to mirror repeated immune infection in mouse models. So just to briefly talk about what LPS is, it's an endotoxin found in the outer membrane of gram-negative bacteria, and it's composed of these four main subunits, and it's been found to produce fever, decrease white blood cell number, and cause damage to blood vessels in humans. So it's, again, repeated LPS injections are commonly used as a proxy for repeated infection in mice. So one such study that did this was this 2018 Wonderland paper. So they basically gave low dose LPS injections peripherally and then assessed for changes in the brain. And the injections were spaced 24 hours apart. So for example, in the 1x LPS group, on the first 3 days, they got PBS and then were given a final injection of LPS, and then brains were analyzed after that. Same for 2, 3, and 4. And they found that after 1 hit of LPS, the immune response and microglial activation increased and then peaked after the second LPS hit. So that was indicative of an immune priming effect. And then after the 3rd and 4th hit, they saw that the immune responses and microglia activation started to tamper down and tolerize. And in this study, they also saw that microglial immune priming was dependent on HDACs 1 and 2. So to respond in such dynamic ways, microglia actually have what's known as a kaleidoscope of different phenotypes that are highly dynamic. So microglia take on various transcriptome and epigenome profiles in various activation states, and also influenced by a wide range of factors, including age, sex, species, experimental procedure, disease. And classically, it's thought that activated microglia adopt an amoeboid unramified form that allows for increased mobility to sites of infection and more efficient cytokine release. However, the relationship between their morphology and function is not so dichotomous and clear-cut as classically thought, and they actually take on a whole range of different morphologies in between the amoeboid to branchy states, even showing some rod-like properties, which is interesting. So we wanted to ask how repeated low-dose LPS stimulation shapes innate immune memory responses in microglia, and we probed this question from these 3 angles. First, looking at the mouse's sickness behavior, profiling their cytokines in the brain, and then looking at microglial morphology. So I'll start with the first. We use that same paradigm that I just discussed a few slides earlier with low-dose LPS hits in just wild-type mice, and then assess sickness behavior 3 hours after the final injection in each of these groups. And the sickness behavior was scored on a 3-point scale, assessing for parameters like lethargy, ptosis, and piloerection. And the scale ranged from 0 to 3, with 0 indicating no symptoms and 3 indicating all symptoms. And we saw that after 1 hit, the sickness behavior increased and then peaked after the second hit of LPS, and then started to tolerize back down after the 3rd and 4th hit. So to recap, there was a peak in sickness behavior after 2 hits of LPS that mostly resolved by the 4th hit. Then we looked at cytokine profiles. So to do this, we used that same model that I went over, and instead of assessing for sickness behavior this time, we actually sacrificed the mice and then collected their brains to look at 3 hours after the last injection, took half hemispheres, and dissected out various brain regions. So the frontal cortex, which is important for cognition and decision-making, striatum, involved in reward and motor systems, hippocampus, involved in learning and memory, and then the cerebellum, which is important for motor learning. And then we took those microdissections and isolated proteins out and assayed for a panel of cytokines and chemokines using the Luminex platform. And before I delve into the specific results here, I'll just briefly go over how to read this graph. So on the x-axis, you have comparisons between your control and each of the LPS conditions. And then on the y-axis is the log fold change of the picograms per mils of each of the cytokines paneled. And these are all split up by the different brain regions that we looked at. So from the panel, amongst the growth factors, we saw specifically, or I guess I'll just point to specifically GCSF. In the brain, it's been shown to control proliferation and differentiation of neural stem cells, and been seen to be important for glial cell activation. And then in the pro and anti-inflammatory cytokines, we saw IL-6 specifically, which has been shown in multiple studies to be important for brain function and disrupted in brain disorders. Saw that there was a sensitization at the second hit that either resolved or sort of stayed up, at least in the striatum. And then looking specifically at chemokines, we also saw a handful of them that were significantly upregulated after the second hit and either resolved back down or stayed sensitized after the third and fourth hit. The main take home from this is that across brain regions, we saw an increase in cytokine responses after the second hit of LPS that either, or for the most part, tolerized by the third and fourth or sort of stayed up, which is interesting. And finally, for the microglia morphology, same model and dissection, we took half hemispheres. And for this, we started with the frontal cortex as our first target brain region. And we stained for a microglial specific marker and then used ImageJ analysis to look at their skeletons. So that involved picking out our region of interest, in this case, the frontal cortex, looking at the microglia, binarizing the images, cleaning up any debris particles, and then thresholding the specific cells, skeletonizing them, and then analyzing them for different things like endpoints, branch lengths, and junctions, which give you insights into different parameters like cell ramification and cell branching complexity. So we saw that in terms of maximum process length, which was used as an indicator of cell ramification, that there was an increase at the second hit of LPS that sort of maintained at the third and fourth. And in terms of branching complexity, we also saw an increase in the number of endpoints at the second hit that also seemed to maintain at the third and fourth. This is also seen in the other measures of branching complexity. And these are images taken from the, or little mini images taken from the actual fluorescent images, where we saw that at PBS and 1x LPS, the microglia seemed less branchy and less complex, but then they sort of hyperramified after the second hit and seemed to stay that way at the third and fourth. So to summarize, across measures, there seemed to be a peak in sickness behavior, cytokine profiles, and microglia morphology at the 2x LPS hit that either mostly resolved by the fourth hit or maintained by the fourth hit. And for future directions, I, at least specifically for the morphology, would want to assess some more dimensions like cell soma size and thickness of the processes, as well as identify any candidate genes and epigenetic regulators that are driving the changes seen across these parameters. And we've started to do a little bit of that, looking at gene expression profiles as well as gene regulation, which Dr. Ciernia will touch on in her talk. With that, I just want to say thanks to all my wonderful lab mates and the support system that I have at UBC and through the grad program in neuroscience and funding that I get from CONP. Thank you.
Thank you very much, Jennifer, for this very interesting presentation. I'm going to check if we have any questions. So there's a question from Alina Rasul. Is there a research paper we can refer to?
A research paper like for our study specifically?
Yes, I guess.
No, these are all preliminary results, so stay tuned.
Okay, so I have a quick question about the future directions that you're taking. So do you have any specific epigenetic profiling in mind that you're going to address, or is there any known epigenetic modifications that are involved in these sickness behavior process, or that would regulate the morphology, for example?
So there's a whole host of studies looking at repeated LPS, but they're all sort of done in slightly different ways, where it's various doses of LPS, the spacing is different, and there's some people see priming, some people see tolerance. But amongst those studies, there are some epigenetic marks that people have profiled. Like in the Wendelin paper that I discussed at the start of the presentation, they looked at, or they knocked out HDAX 1 and 2 and saw that that countered the effects that they would see in immune priming. So that's a candidate target. There was another paper that looked at a lower dose at just one hit of LPS and saw that there were changes specifically at the IL-1 beta cytokine promoter, looking at like H3K9 and K14 acetylation. So that might be another candidate target to go after.
Okay.
There are many out there.
That's good, thank you. We have a few more questions that came up. So Carolina Trupini has asking, great talk, beautiful slides, Jennifer. How generalizable do you think these responses are to other types of insults beyond LPS?
Yeah, so I'm gonna go back to one of my former slides here. Yeah, so at least with respect to morphology and all of these other factors, they see that it seems to be stimulus dependent as well. So effects that you see in response to LPS might not necessarily translate to another type of stressor. So the studies that we at least use to inform our work, we try to look for like LPS specific models. But I know like this classic branchy to amoeboid form as a indicator of activation is not seen in a depression model where they saw hyperramification of the microglia processes in response to stress. So that's why it's sort of really important to probe these questions from all these different angles, which is what we're trying to do in our work.
Okay, that's very good. We have a question from Anna Nikolic. Do you see any reorganization of chromatin on immunofluorescence upon microglial activation? For example, by high resolution DAPI?
I haven't actually looked at that yet, but we did stain for some epigenetic marks and I'm in the process of analyzing it. So I don't have any answers for you yet, but that work is coming and Dr. Ciernia will also touch on epigenetic regulators of this in her part of the talk. So.
Okay, very good. We have a question from KJ Aitken. Do the persistently upregulated cytokines, KC for instance, have a role in regulating the morphological changes that you are seeing persistently?
I did look into the cytokines and I couldn't really find any, get to the slide. Yeah, I couldn't really find anything specifically related to morphology. But I do know that especially the ones that I highlighted throughout my talk have been implied in brain function and seen to be disrupted in brain disorders and also like blood-brain barrier integrity and that type of thing. So not morphology specifically, but they do seem to be important for the brain.
Okay, so we'll take maybe one last question from Karine Doiron. Does LPS cross the blood-brain barrier? And will you be looking at other types of insult that relate to viral infection in the brain?
So LPS is thought to not cross the blood-brain barrier at such low doses. Though in models like MS, where the blood-brain barrier is compromised, it certainly could. But we are gonna test that as well to look for whether or not LPS crosses the blood-brain barrier. So I guess to answer the question, it's generally not thought to at such low doses.
Okay, thank you very much. So I think in the interest of time, we'll go on to the next speaker. So our next speaker is Dr. Annie Ciernia. Dr. Ciernia joined the University of British Columbia as an assistant professor in 2019. The research interest of our group consists in understanding the epigenetic mechanisms of transcriptional regulation that are involved in brain plasticity and neurodevelopmental disorders. Dr. Ciernia did her graduate work at the University of California, Irvine, under the supervision of Dr. Marcelo Wood, where she studied the role of a neuron-specific nucleosome remodeling complex in regulating long-term memory formation. She then went on and accomplished a postdoctoral work at the University of California, Davis, with Dr. Janine Lassalle, where she focused on understanding how DNA methylation and chromatin accessibility impact gene expression in autism spectrum disorders. During this postdoctoral work, Dr. Ciernia was also co-mentored by the autism in the autism training program at UC Davis, Mind Institute by Dr. Jacqueline Crowley, who's an expert in animal model behavior related to neurodevelopment and autism, and also by Dr. Paul Ashwood, an expert in autism spectrum disorder neuroimmunology. So Dr. Ciernia then joined University of British Columbia in 2019, where she is now an assistant professor in biochemistry and molecular biology in the Faculty of Medicine. Her current work focuses on the understanding how early life perturbation to the epigenome, perturbation of the epigenome alter microglia neuron interaction in the developing brain and lead to lifelong changes in cellular function and behavior. So she will be presenting her work in a talk that's entitled Epigenetic Conductors of Microglia Immune Memory in the Brain. Dr. Ciernia, you can go ahead.
Thank you so much for that lovely introduction. You can see my slides okay?
Yes.
Okay, terrific. All right, so I want to start with a few acknowledgements. First, I want to say thank you to the organizers of the seminar for inviting me to talk. It's super fun to come share our work with all of you, even if it's virtually. This is sort of the next best thing to in person. And I want to start with a land acknowledgement. So my lab is located at University of British Columbia on the Point Grey campus, which is on the traditional, ancestral and unceded territories of the Musqueam people. And I want to also recognize my research group. So this was taken pre-pandemic, but I feel really fortunate to have built a wonderful lab even in such a short time. So I started my lab here at UBC in July of 2019. We were up and running about 6 months. And this was taken just prior to the research shutdown about a year ago, last March. And I feel very fortunate that the crew that I have on board has done phenomenally well under very difficult circumstances. And I'm going to be talking about some of the work that we've been doing together over the last year. And I also want to give a shout out to the Molecular Epigenetics group at UBC. So this is a terrific group of supportive faculty, both senior and junior folks that get together weekly for journal clubs, and then also we meet as a PI group pretty regularly. They've been super helpful in informing the way that I think about my own science as well as my grants. And I really appreciate what a supportive environment that we have here. Okay, so for the main part of the talk today, I'm going to be talking about transcriptional memory in microglia, and specifically how repeated immune activating events alter microglial transcription. And Jen very nicely set this up for me, so I'm not going to do a ton of background because you already saw a lot of the important background for why we're interested in regulation in microglia. And I'm going to focus instead on the impacts of repeated immune activation on gene expression, and then some preliminary work we've been doing looking at epigenomic regulation. So Jen nicely introduced our model system that we've been using in the lab, where we give repeated injections once every 24 hours. And so we have animals that receive a single LPS dose, 2 doses, 3 doses, or 4 doses. And similar to the cytokine profiling that Jen talked about, I'm going to talk about work we did actually in the same animals, but from the other hemisphere, where we looked at gene expression by RNA-seq. So again, we have 3 different brain regions that I'm going to be showing you data from: frontal cortex, striatum, and hippocampus, and our differential gene expression analysis from this. And we had several goals going into this analysis. One was to identify how these brain regions might be responding either similarly or differently to a peripheral LPS injection. So we wanted to compare across regions. And we wanted to see if there was anything common about the different regions or unique that might give us some insight into function. And then the majority of the talk actually be focused on some work we're starting to do looking at upstream transcriptional regulators of some of the differentially expressed genes we've identified. Okay, so first I'm going to show you some data looking at each brain region where we identified differentially expressed genes by comparing each LPS condition to the PBS controls. And so what I'm showing here is a volcano plot, so log fold change by negative log 10 of the adjusted p-value. And this is data for the frontal cortex. And so this is genes that are differentially expressed in the single LPS condition compared to the PBS controls. And what you'll notice is there's only a handful of genes that change in their expression. However, if you instead give the animal 2 doses of LPS, now all of a sudden you have almost 250 genes that get upregulated. And interestingly, if you instead give a 3rd LPS dose, we now see a blunted change in gene expression. So we're back almost down to the 1x levels, and similarly for the 4x. And we've looked at this now across other brain regions. So this is the hippocampus, which looks very similar. So you have this increase mostly limited to the 2x LPS condition. And similarly for the striatum. One interesting thing that came up in the striatum that is unique to this brain region is we also see some genes that are increased in response uniquely to the 4x. And we're interested in pursuing some of those in the future. And so if you compare across these brain regions, and I'm just highlighting this with this box, the 2x LPS condition, this is really where the action is at in terms of the increase in gene expression in response to LPS. If you look at this a slightly different way, so it's the same data shown in a heat map where we've clustered, each row is a gene, and we've clustered the genes by the pattern of expression. So whether they're increased or decreased in response to the different LPS treatments. And if you look across the different brain regions, the patterns actually look somewhat similarly. And we find the most differentially expressed genes in the frontal cortex compared to the other regions. But what I really want to highlight is what's shown in these pink boxes, where we see this consistent pattern of gene expression, of increased gene expression in response to the 2x LPS that then tolerizes back down into the 3rd and 4th. What was interesting is if you compare these genes inside these pink boxes for the 2x LPS, which is what's shown here in this Euler plot, they actually are largely unique. So even though they're regulated in the same pattern, they're not necessarily the same genes. So there's actually only 6 that are shared across all 3 brain regions. And we are interested in some of these, but it was actually quite striking to us that even though these patterns of expression are the same, they're not necessarily the exact same genes. And so what I just showed you is that we have these differentially expressed genes in response to the 2x LPS condition, and that they're mostly unique across brain regions. So we then wanted to say, okay, even though we have unique genes, maybe there's some common downstream regulatory pathways that might be impacted that might hint at some shared function. And so we did some pathway enrichment analysis of the differentially expressed genes. And we identified pathways that are enriched specifically for genes that are upregulated in response to the 1x, 2x, 3x, and 4x. I don't expect you to read any of these. The take-home message is that even though the underlying genes aren't necessarily the same, many of the pathways are highly shared. And so we see canonical immune response pathways for things like viral responses, nod-like receptor signaling, interferon gamma. And we also found this potentially interesting adenosine receptor signaling. So there is some work suggesting adenosine regulation by microglia can impact neuronal function. And this is one of the pathways we're interested in trying to look at in the future. And so in general, this supports a role for immune cell regulation as the primary target of LPS, which is perhaps not surprising considering you're giving an immune activating event. In parallel to our differential gene expression analysis, we also ran some weighted gene network analysis, so WGCNA, which is done completely differently, but also found basically the same finding. And so we identified this module of gene expression that was significantly related to our LPS treatment. So what I'm showing here, get my cursor back, is frontal cortex, hippocampus, and striatum for each one of the treatment conditions. And you can see these interesting clusters of increased gene expression in response to the 2x. This is a little easier to see in a box plot, which is the same data just collapsed down. So you can see that in response to the 2X, you get this nice increase in gene expression, which was a nice validation for our differential analysis. And again, we see very similar types of pathways enriched within this module, things for NF-kappa B signaling, adenosine, and enrichment specifically for microglia and other cells of the blood-brain barrier. We've since gone in and started to validate some of these different genes. So I'm just going to show you one example that actually Jen has been working on. So this is a chemokine, so CXCL16. And this is how the gene is expressed in our RNA-seq. So we see an increase across basically all 3 brain regions. This one's not quite statistically significant, but it increases basically in response to 2X LPS and then tolerizes back down. In a completely different set of animals that was run about a year after the first one, we see something very similar. So an increase in response to 2X that then tolerizes back down. And one of these genes, why are we interested in this gene? Well, it was one of the top differentially expressed genes. It's also highly expressed in microglia in the brain. So this is data from a published database showing expression in different cell types. And you can see this big preferential expression in microglia. And we've started isolating microglia out from the brain. So this was an experiment we did where we're actually isolating microglia from animals that had received either 2X, 4X, or PBS. And you can see pretty much the exact same pattern. So this increase in response to the 2X LPS, indicating that at least a subset of the genes that we're seeing in the RNA-seq, their expression might be driven by changes in microglia. So I've talked a little bit about these downstream common regulatory pathways that are impacted, including things like immune pathways, adenosine, and metabolism. But what I really want to focus the rest of the talk on is the work we've been doing more recently looking at upstream transcriptional regulators. And I'm going to start this by going through sort of what our working model is for epigenetic regulation of priming intolerance. And so this model comes primarily from work done in other peripheral macrophage populations. So every resident tissue in your body has a uniquely adapted set of resident macrophage immune cells. Microglia are a bit unique in that when they adapt to their local environment in the brain, they're not replaced by the periphery. And so they locally adapt their epigenome uniquely to the brain. And so this is sort of our basic working model that we're going to be testing, but it doesn't necessarily mean that this is what is happening in microglia. And so the working model for this is that under sort of homeostatic conditions, so normal healthy brain, microglia keep pro-inflammatory gene expression low because you don't want to be activating an immune response unless you actually need to respond to a pathogen or an injury. And to keep these genes in a low baseline state, we predict low levels of acetylation and activating methylation marks at enhancers and promoters. And it's been shown, at least in peripheral macrophages, that RNA polymerase II is often paused at the start site of many of these LPS responsive genes. In response to LPS treatment, we predict that histone acetyltransferases and methyltransferases will add on activating marks to both enhancers and promoters. That then in turn recruits in chromatin remodeling that allows for opening of the chromatin environment that facilitates transcription factor binding and potentially looping between distal enhancers and promoters. And that really this sort of setup state then leaves you in this primed responsive state, such that when you give a second LPS dose, you can then more rapidly release and engage transcription, producing that increase that we saw with the 2X LPS treatment. The flip side of this is what we predict might happen in the case of tolerance, in which you have to remove those activating marks with the histone deacetylase or demethylase. You then potentially need to recondense your chromatin environment. The loops may or may not disappear depending on probably the specific locus that we're going to be looking at. And this type of repressive environment, we think, might lead to the repression of gene expression that we see in response to the third and fourth dose of LPS. So this is the working model kind of in summary, where you have this baseline state that then becomes primed for activation. And then there's some sort of switch that occurs that allows it to turn back into this sort of resistant state that we see with tolerance. And so we were developing this model about a year ago, and then before we could get into the lab to start testing any of this ourselves, we ended up in COVID lockdown. And one of the fortunate things that happened around this time was that this paper came out from Stefan Young's lab, looking at IL-10 and its role in response to a single dose of LPS. This is a really great paper. I highly recommend it as a read. One of the things that they did that they didn't really analyze very much was they did some attack-seek in response to LPS. And this was like one figure in the supplement buried. And so it wasn't a main aspect of their paper, but it was super interesting for us in that they give a single dose of LPS and then they collected microglia from the brain at 12, 24 hours, and 7 days after that injection. And then they took those microglia and did attack sequencing on them. And so what we did actually during the COVID lockdown was we did a bunch of bioinformatic training with the people in my lab, and we started to analyze some of this data for our own interests. And so we actually called differentially accessible regions between PBS and each one of the different LPS conditions. And then we looked at how these differentially accessible regions clustered based on their pattern of activity, as well as whether they were within a promoter or within a distal enhancer. And so this is the data that I'm going to show you next. And so just as a reminder, we're looking for regions that potentially are increasing in response to LPS. That's the model that we're predicting. And so do we see this increased accessibility following LPS treatment? So I'll unpack this a little bit for you. So what we're looking at is the changes in chromatin accessibility at the promoters of genes in response to 1x LPS. And so if you focus here, we're looking at the read depth normalized attack signal. So basically how open are these regions? And then we clustered these different regions by their pattern. And so what's shown here in this heat map is each individual condition. So this is the vehicle 12 hours after LPS, 24 hours after LPS, and 7 days. And so for cluster 1, for example, they're not that open in baseline, and then they open transiently in response to LPS. You can see similar patterns for some of the other clusters. So in this case, the promoters are slightly open, and then they significantly open and stay open. This is especially prominent for cluster 3, where they're basically open all the time, and maybe even stronger by 7 days, and also for cluster 5. So these changes between the different clusters are relatively subtle. Most of them have at least some accessibility at baseline and then increase in response to the LPS treatment. But what we wanted to know is, are these changes in accessibility over the promoter linked in any way to the differential patterns of gene expression that we've seen? So this is a little schematic of this. Do we actually see this increase in promoter accessibility, specifically over genes that show this primed phenotype? And so we took our differentially expressed genes. So this is what the heat map I showed you before, where each line is a gene. And then I lined this up against the promoters and their accessibility. So this heat map is how accessible these promoters are at vehicle conditions in response to 12 hours, 24 hours, or 7 days. And what you can see, just as we predict, is that the increase in promoter accessibility goes up in response to LPS, very consistent with this idea. However, if you then look at all of the genes that are differentially expressed, they actually all show this pattern. And so they are relatively closed at baseline compared to how open they become in response to LPS, regardless of their pattern of expression. And so there's nothing that we can identify at the promoter in accessibility, at least, that predetermines genes that will respond uniquely in this 2x type of pattern. And so there's a lot of reasons for this, potentially, including that promoter accessibility isn't sufficient to actually look at priming, that is actually transcription factor binding or something else at these promoters. Or maybe we're looking in the wrong spot. Maybe promoters actually aren't the best place to look at in terms of dynamics for regulation of these differentially expressed genes. And so the next thing that we did was we moved on to look at the enhancers. So these are regions that show differential accessibility in response to the 1x LPS, but they're not within 3 kb of the transcription start site of any gene. So they're outside of genes. And we've called them enhancers. They're putative enhancer regions. And again, I'm showing you the read depth normalized attack signal for each one of the different clusters that we've identified. In the vehicle condition, for example, for this cluster that gets repressed, they're slightly open at the beginning and then they close in response to the LPS treatments. We also see other types of patterns, so delayed opening, where they're somewhat open at baseline, and then the strongest accessibility is actually 7 days later. These interesting sustained cluster that opens and stays open, and we're particularly interested in some of the regions that underlie this cluster, and ones that transiently open and then close. And so we wanted to then ask, okay, so we have these regions that show differences in accessibility. Are there any other epigenetic signatures that might help explain why these different clusters have different types of dynamics in response to LPS? And what we did to start to address this is we went into some of the published data sets that are available for isolated microglia. So all of these data sets are at baseline. There's no LPS treatment. So it just gives us some insights into what might be regulating these specific enhancers at baseline. And we looked at H3K27 from David Goslin's work, H3K9 acetyl, H3K4 dimethyl, H3K27 trimethyl, PU1, which is a lineage determining transcription factor important for microglia regulation and development, and then RNA polymerase 2. And so we downloaded all of this data from Geo, realigned it all and processed it in the same way so that we could compare it across all of these different methods. And what I'm going to show you is what each one of these different signals looks like over our different enhancer clusters. So the first one I'm going to start with is this H3K27 acetyl. And what we see is a nice signal over these delayed, sustained, and transient putative enhancer regions, indicating that especially for the sustained, these might be active enhancers. We do not see much for H3K9 acetyl over these regions. We do see some signal for H3K4 dimethyl, again, indicating that some of these enhancers might be active. We don't see much for the repressive H3K27 trimethyl mark. We do see nice PU1 binding, especially at these sustained enhancers. And we see maybe a little bit of RNA pol 2, indicating that maybe some of these enhancers might have some eRNA transcription, but nothing that was super outstanding. And so this leaves us with sort of this model from what we can glean from publicly available data, where we have increased acetylation and methylation indicating active enhancer usage, and then also PU1 binding at some of these regions, specifically at this sustained cluster. So we then wanted to ask, okay, is there anything that we know about what types of epigenetic regulators might be laying down some of these specific marks? And there's been exactly one paper showing that if you delete histone deacetylase 1 and 2 in microglia, you can drive phenotypes, including changes in microglial development and reactivity during aging and Alzheimer's disease. And this particular paper had some H3K27 and H3K9 acetyl ChIP-seq from the HDAC1 and 2 knockout animals, where they have HDAC1 and 2 missing only in microglia. And so we then took those data sets and looked to see what the levels of the acetylation signals were within each one of our different clusters. And so in blue, you can see the H3K27 acetyl for the wild type. And then in red is H3K27 for the HDAC1 and 2 microglial knockout. And what hopefully is very obvious is that you have this big increase in signal in the knockout, specifically mostly over this sustained cluster, indicating that when you lose HDAC1 and 2 repression, you might be releasing repression of these enhancers, allowing them to activate potentially inappropriately in the knockout. However, this appears to be unique to H3K27 acetyl, as we don't see any differences between H3K9. If you go ahead and quantify this, so this is the exact same data just shown as a log plot of the signal over each one of these regions. we actually have significant increases in H3K27 only within this cluster 4. This is almost significant, but this is the main impact that we get with loss of HDAC1 and 2 in microglia, really indicating that HDAC1 and 2 might be targeting this region to have baseline repression in response to homeostatic microglia. And so HDAC1 and 2 might be required for the initial baseline repression of histone acetylation at some of these LPS inducible enhancers. We then wanted to ask, okay, so we have some candidates for looking at histone acetylation regulation. Are there other potential transcription factors that we should be interested in besides what we've looked at so far with PU1? To address this question, we looked for enriched transcription factor binding site motifs within our different enhancer clusters. And so what I'm showing you here is the number of shared motifs across each of the different clusters. So clusters 2, 3, 4, and 5. I'm just going to highlight a few things that I want you to take home from this. So the majority of the motifs that we found were actually shared by these two clusters 4 and 5. They had motifs for the ATFs, as well as FOS and June, some Krebs activating sites. So pretty standard activating transcription factors. If we look at sites that are shared across all of these different regions, we have interferon response elements, PU1, and some other immune activating transcription factors. We also see in clusters 3, 4, and 5 that have dynamic increases in response to LPS, several of the known microglia transcription factors, including F2C, RUNX1 and 2, that are known to be important for microglia function and activation. We also see some unique things. So in this transient cluster is where we find NF-kappa B binding, as well as HIF1 alpha. The delayed cluster has some other types of transcription factors, including SOX and SMAD. And one of the ones we were actually the most interested in was trying to identify transcription factor binding motifs that were unique to this particular cluster where we see sustained activation. And one of the ones, there's only 4, and one of the ones that we pulled out was this NFAT, which was particularly interesting because this has been previously linked in microglia to signal-dependent transduction in response to LPS, and has been found to mediate phagocytosis or microglial activation state. So we're interested in pursuing that one as a potential interesting upstream target that might be regulating some of these sustained enhancers. And so we then wanted to ask, is there any way we can link these potential enhancers back to their target genes? And so there are a number of different strategies for doing this. The easiest one is just to pick the enhancer with the closest promoter and link those two together. But that is actually probably not the best plan. And so I'm showing here some data from Alexei Knott's work a couple of years ago, looking at human microglia in the brain, and they did plaque-seq or HiChIP, where they were able to link microglial promoters to specific enhancers. I'm showing here our favorite gene, CXCL16, and it actually has a promoter and enhancer pair that is relatively close in space, but the vast majority of their linkages actually were not the nearest promoter enhancer pair. And so we're interested in potentially generating some of this HiChIP data for microglia so that we, in the mouse, in response to LPS, so we can actually start to causally link some of these enhancer promoters together. In the mouse, so this is the exact same data, but in the mouse, we have this putative enhancer region just upstream of CXCL16. And so we're interested in trying to test this as proof of concept that there might be a link between this potential enhancer region and our gene of interest, similar to what you might see in human. And so this leaves us with the model that we're still working on, that in response to the first LPS treatment, you get these increase in H3K27 acetyl and some of the methylation marks. And that is potentially involved in recruiting some of these transcription factors and potentially in creating some of these loops or modulating their activity. And that in response to the second LPS treatment, you get a rapid induction of gene expression that potentially triggers this onset of tolerance that you see in response to the third and fourth, and that this may involve some of the HDACs to remove some of these epigenetic signals. And that then in response to the third and fourth LPS stimulus, you actually see repression of gene expression. And so this brings me back to the sort of outline. And so I showed you some data looking at dynamic enhancers that open in response to 1x LPS and some potential transcription regulators upstream of those processes, and speculated a bit on what might be leading to tolerance. There's far less known about that than about priming. So this brings me to the conclusions. So 1x LPS appears to prime gene expression, and then in response to 2 and 3x LPS, you get this tolerization. Brain regions respond differently to these repeated LPS, but actually the downstream pathways appear to be quite similar, or at least conserved. These LPS sensitive genes may be regulated by these distal enhancer regions that show high levels of baseline acetylation, and that enhancers with this sustained accessibility pattern might be targets for PU1 and NFAT regulation. And then to tie this back into some of the work that Jen showed you, this microglial priming of gene expression really happens at multiple levels. So we see changes in gene expression, sickness behavior, cytokines, morphology. I didn't show you any of our phagocytosis data, but we also see activity responses. And so really we're trying to hit this from multiple different angles to understand how the gene expression is regulated and how that ultimately translates into function and changes in the actual animal. So some of the things that we're going to be doing moving forward is to actually look at histone acetylation in response to repeated LPS, and then try to link those to their target genes. And we're interested in trying to look at some of these mechanisms of tolerance, which is far less well understood, and trying to look at some of the histone acetylation regulators that might be inducing tolerance, or whether it's a different epigenetic regulator. And then how do these primed and tolerized genes actually change microglia function? So we have yet to actually link these differentially expressed genes directly to things like phagocytosis function or a specific change in morphology. Of course, the ultimate goal is to try to figure out how this works, so you could potentially block priming or induce tolerance in disease cases like Alzheimer's or aging, or even neurodevelopmental disorders where these pathways and processes seem to be disrupted. And so with that, I'll be happy to take any questions. And I just want to thank all of the funding that we have been so lucky to receive over the last couple of years.
Thank you very much, Dr. Ciernia, for this very excellent talk. It was really, really interesting. I'm going to start maybe with one question and then I'll move on because I see questions coming up in the chat. So I was very intrigued by your metabolic profile change, especially the adenosine metabolism that comes up on LPS. And I was wondering if you would expect the LPS to alter the metabolic profile of the microglia, especially because adenosine is linked to pathways that could regulate the availability of SAM, for example.
Yeah, absolutely, 100%. And I didn't emphasize any of that, but actually, so Dr. Brian McVicker is just down the hall from us, and they have some beautiful work showing metabolic reprogramming in microglia in response to LPS. So that's something we're certainly interested in exploring, possibly with them, to actually try to link our gene expression changes to those functional reprogramming and metabolic usage. So yes, that's definitely something we're interested in. And the link between microglia and neurons is also interesting. So microglia can help regulate adenosine release that can impact neuronal function. And Ann Schaefer's lab has some beautiful work over the last couple of years looking at microglia neuron crosstalk and how that might be important for sort of keeping neuronal activity in check. And so when you mess with that system, you actually predispose the animals to epileptic seizures. And so we're interested in trying to understand that also in terms of autism, where seizures are also quite common.
Okay, that's very interesting. Thank you. I'm going to go to the questions. So there's a question from Carolina Tropini. She says, really great talk and interesting talk. Annie, CXCL16 regulation for the 2 dose of LPS has this interesting responder, non-responder phenotype. It looked like 2 of the mice had increased expression and 1 looked like a control. Is there something else different about the non-responders in expression or other genes or any other phenotype that separates the two groups?
Oh, that's a really good question. Yeah, so I mean, these are always variable, right? So you're in vivo in mice, you're injecting LPS, and it's actually surprising sometimes to me that we get as consistent responses as we do. Yes, there are definitely some variability, and we haven't gone in on an individual animal and started to parse that, but we could now. So Jen has done like 5 or 6 different cohorts with various different manipulations, all sort of variants on the same paradigm, and we could go in and look at the CXCL16 expression and compare it at least to sickness behavior, because we have that from every mouse. So we score them as they go along to make sure that our LPS injections are good and the animals show the correct behavior. That would be interesting to do though, and because we do see variability in the behavior response also. Yeah, I'll do that and get back to you.
Good, thank you. I'll go to the next question from Jim Davey. Great talk. Have you looked at a role of mitogen and stress-activated protein kinase, MSK, and H3S10 phosphorylation or H3S28 phosphorylation and LPS-induced expression?
Yeah, those are both great questions. And no, we haven't yet. We haven't really gone into the upstream signaling cascade at this point. What we have done is done a little bit of preliminary work looking at NF-kappa B localization, which doesn't seem to be impacted by repeated LPS. So we think the level of regulation is not necessarily at the level of signal transduction, at least in our hands in this model, for the one thing we've looked at. So we have a bit of work to do to look at that, but we haven't started to probe those upstream things, but that's a great suggestion. And it's possible that some of the tolerance and priming might occur not only at the epigenetic level, but also at the level of signal transduction.
Okay, thank you. I'll take another question from Charles Villarino Guel. Very interesting talk. Did you assess which NFAT factor was involved in the activation? It would be interesting if it was NFAT1, NFAT C2, as it carries pathogenic mutation for multiple sclerosis.
I was unaware of the MS link. That's super interesting. No, I'll have to go back in and see if the motif that we pulled up is more similar to 1 versus 2. I assume that they're potentially different. I didn't consider that. No, but that's a great suggestion. And I was unaware of the link to MS. That's super helpful. Thank you.
Thank you.
It looks like, does blunting tolerance happen with longer gaps between injections? And do you think the multiple injections represent repeated infections or physiological crosstalk with inflammatory microbiota? Yes, great questions. The blunting itself, we've done one experiment where we've looked at the long-lasting ability of priming. And Jen did this, at least preliminarily. It looks like priming lasts at least a week for some genes, maybe not for all of them. We have not tried to do this with tolerance yet, although I think that should be on our list of things to do, is to tolerize and then maybe look a month out. What I can tell you is that, at least for the work that the Marco Prince's lab did in the Wendelin paper, they did either 1x LPS or 4x, and then they looked 6 months later and saw impacts both on gene expression and on microglia function, indicating that some of these impacts, whether they're direct or indirect on microglia function in the brain, seem to be quite long lasting, but we haven't done anything that long yet. But if we want to understand, I think, some of the potential therapeutic implications of tolerance induction, I think that's something that we should start to play with. And as far as the microbiota goes, yeah, great question. Not as sure in the adult how much it might impact. We're also doing some of these early life priming things in early life, so as more of a neurodevelopmental disruption model. And in that case, I think the microbiota might be super important. So that's something that we could certainly start to play with by either looking across different vivariums or profiling our animals and see what they actually have.
Yeah, so I think now everyone is unmuted, so you can raise your hand and ask questions. But if I can just ask a quick question, maybe I didn't get it, but was your experiment done on mice, female or male mice, and does it make a difference?
Yeah, so that's a great question. So most of the data I showed you was from male mice. So the RNA-seq was in males. We have done this also in females, and at least for most of the gene expression, the patterns seem similar. So like CXCL16 looks similar. Maybe the magnitudes are slightly different, but the patterns are similar. Jen has started to do some morphology work. So she has some data from basically the flip experiment from what she showed today, which was in males, also in females, and they are a fair bit different. So we're still trying to parse some of those sex differences and to try to figure out what's important for function, what is just sort of phenomenology. But we definitely predict that there will be differences, but maybe not in every measure that we've looked at.
I might have one last question. Would you, if you don't mind,
sure,
would you expect DNA methylation to be affected during that process?
Yeah. So actually, all of my postdoc was mostly on DNA methylation, and one of the things that I'm thinking about doing is to look at DNA methylation, not so much in these early dynamics, but sort of in the long-term lockdown of this phenotype. So in perhaps this long-term tolerance, my guess is actually that histone acetylation will be early important for sort of the setup, and then methylation will actually lock that state in. Okay. So that if we start looking at some of these long-term time points, that would be perhaps when we want to look for methylation changes. But yeah, we just haven't gotten to those experiments yet.
Okay. Very interesting.